Let's try to understand neoplasia. So neoplasia is just new growth which is highly unregulated and highly irreversible. Okay, you cannot reverse this, and it's monoclonal. It comes from a single cell, right? Let's try to build our concept on this thing called monoclonality, right? Neoplasia is just new, abnormal, uncontrolled, irreversible growth of cells, right? It's unregulated because the cell grows without any normal control mechanism, right? And it's irreversible. This is irreversible because once the cell starts becoming neoplastic, they cannot go back. They will not go back, even if the stimulus is removed. And monoclonality, all the cells in the neoplasm, they come from a single parent cell that has acquired the mutation. So what is clone? Group of cells that derive from one single original cell, and that DNA rearrangement or that mutation gave rise to the monoclonality. Okay, now this monoclonality is the fingerprint of true neoplasia. Okay, if there is neoplasia, there is monoclonality. On the other hand, on the contrast, hyperplasia, it's polyclonal, means many normal cells, they proliferate. Example, hyperplasia of the uterus during childbirth, right? So what does this really monoclonal means? Well, in our body, every tissue has many cells, right? They come from different progenitor cells, right? And when you stimulate these cells, for example, estrogen stimulates endometrium, right? So what happens is that this estrogen will stimulate endometrium, and then endometrium will make many clones, right? This is called polyclonal. But when one cell is genetically messed up or genetically mutated, it gives growth advantage to that one single cell, and this one single cell multiplies uncontrollably, and this all daughter or son cell, whatever you want to call them, all these cells, they carry the same genetic fingerprint of that monoclonal. Okay, so for example, this is polyclonal example. This is one tissue. These are 1, 2, 3, 4, 5. So these 5 cells will give growth to all the clones, right? So the ratio of polyclonal will be 1:1. How? Okay, I'll explain you. Now, how do we detect this? We have these genetical markers for this monoclonality, right? Now these genetical markers, we can distinguish between the different cell lineages. Okay, we use something which is called X-linked markers. Okay, we use these X-linked markers, and we can use these immunoglobulin gene arrangements, right? Or these light chain restriction. Okay, so these are a lot of words I just told you. Let's start with one by one. First, I told you about X-linked markers, right? X-linked markers means G6PD or isoenzymes test. Okay, G6PD or isoenzyme test. Okay, now these G6PD gene, so it's a gene, G6PD gene, it stays, it's there on the X chromosome. Okay, X chromosome, we have two chromosomes, X and Y, right? So G6PD, it stays on the X chromosome, and there are like multiple isoforms of this G6PD genes. Okay, they can be A, they can be B, they can be C, like the multiple. So what happens is that in females, this one X chromosome is, for example, randomly inactivated, right? It's randomly messed up, right? And so what will happen is that these cells will express, for example, one type of isoform, that is A, but they will not express B. That's what I'm trying to say. So just what I'm trying to say is that in females, one chromosome has randomly inactivated, right? Random silencing of X chromosome, right? And for example, right here, I'll give you an example. In a normal tissue, right? In a normal tissue or a hyperplasia, the normal ratio of these isoforms of G6PD will be 1:1. Okay, so I just told you we have two type of isoform, A and B. So A will give 50 copies and B will give 50 copies. So 50 copies is to 50 copies. The ratio comes up at 1:1. Imagine if there is some sort of mutation happening at here, neoplasia is gone. So what will happen is that A to B ratio of the isoform of this G6PD, A will produce 50 copies, but B will be silenced. So B might as well just give you like 1 copy. So the ratio will come to 50:1. So that's not 1:1, right? So any deviation from this ratio 1:1 highly suggests monoclonality, and monoclonality is the strong evidence of neoplasia. That's what I'm trying to explain here. Okay, so in neoplastic tissue, if the neoplasm arises from one cell all the cell will have same isoform, okay? The ratio will be deviated, the ratio will be changed. Example, all will be A or all will be B. That's what I'm trying to say. So deviation gives, deviation from this ratio. Ratio means that all cells are contributing 1 to 1, right? For example, we have 5 cells, 1 2 3 4 5, right? A B A B A B A B A and B. So this will give 1A, this will give B, will give 1A, 1B, 1A, 1B, 1A, 1B, 1A, and 1B. 1 2 3 4 5. 5A is to 5B. The ratio is 1 to 1. That's what I'm trying to say. But in neoplasia, what will happen here is that the B will be silenced. We'll have just A. 5 is to, maybe we'll not get any. So the ratio will change dramatically. So that's what I'm trying to get to. Okay, that is neoplasia in simpler words. Okay, that is just monoclonality. Okay, so that's the concept right there. So G6PD or androgen receptor isoforms, right? This is one of the way to understand monoclonality. Another example is to Ig light chain phenotypes for lymphomas. Okay, now the story here is that each B cell, right, produces one type of antibody, right? And every antibody has 2 heavy chains, right, and 2 light chains. Okay, so the concept is that, let me write the concept. It's a really simple concept because, so the concept is that each B cell gives one antibody, right? One type of antibody. And this one type of antibody, every antibody has 2 heavy chains plus 2 light chains. Okay, that's the concept right there. So here, 2 heavy chains can be, mu, gamma, alpha, these type of, and these light chains can be kappa or lambda. Okay, but they can never be both together. They can never be this kappa, or it can be a lambda, but they can never be together. That's the concept here. So in normal lymphoid population, you will have many B cells, right? And some will make K and some will make lambda, right? But what will happen if we have some sort of, messed up situation, right? What will happen? What will happen is that this random shit, right? Randomity of this beta cells, sorry, not beta cell, B cells, where you randomly decide either make K or make lambda, will be messed up. Either there will be K or will be like lambda, right? It'll just be making one type of light chain, right? Now, if B cells become carcinogenic, right, or maybe cancerous, lymphoma or myeloma, right, for example, it will produce one type of light chain, that is kappa or lambda. So only one. So the ratio will be messed up, right? Ratio will become like, for example, kappa is being made 1 is to 20 lambdas. So that's the ratios now. The ratio is messed up right now. It's not 1 to 1 now, right? So that's what I'm trying to say. That's what I'm trying to go. Okay, so again, the ratio of these light chains, right? Kappa is a light chain, right? So that's what I'm trying to say. Okay, so how's the monoclonality determined light chain? Okay, the ratio of K to lambda light chain is 3 to 1. If the ratio is different than this, it suggests of monoclonality. K to lambda means we have 3 Ks that are made by these B cells, 3K and 1 lambda. This is normal, right? So for every antibody, our B cells make 3 kappa and 1 lambda. But if the B cells become cancerous, what will happen that we can make like 20 kappa or plus is to 1 lambda, or it can become something else. Like the ratio doesn't stay the same. That's what I'm trying to go to. Okay, that's the concept right there. Okay, so other thing is, what are the differences between benign and malignant tumors, right? So benign is that they are localized. They do not metastasize, right? They do not have that potential to metastasize. That's why they are benign. Malignant is that there's just one word. They have the potential to metastasize. Even if they have not metastasized, right, even if they are just locally present but have the ability to metastasize, we call them malignant. So they invade locally, have potential to metastasize, don't have to already be metastasized. They are slow growing, benign. They are distinct, means they are well regulated. They are mobile, it means you can move them. And here on the benign point, they have rapid growing because they are really malignant. They want to grow fast. They are infiltrative, means they really want to attack all of your lymph nodes and all of your, just they're really attacking, aggressive. That's what infiltrative here means. And they're fixed to surrounding tissue. It means they are uh, you cannot move them. That's the point. You cannot move them physically with your hands, or maybe with your fingers. Now instead of really you know thinking why it happens, I mean, what people generally do is just they right away just remember that they're fixed to surroundings. Why they are even fixed? That's a good idea, right? Now the idea behind is that why they are mobile is that benign tumor, they like to expand but they do not invade. Okay, that's their idea, the benign, right? That's the idea. The mechanism behind is that they grow really slowly, the benign ones, and they compress the surrounding normal tissue, right? And they have this capsule, this fibrous capsule around them, and this fibrous capsule is well circumcised. It's really mobile. That's what I'm trying to say. So it's like really, it's like a ball inside a plastic bag, and you can roll it easily. This benign thing, and I can give you an example. For example, fibroadenoma, right? This type of cancer is really mobile. It's really well defined. It's encapsulated, right? So it's like a ball in a plastic bag, right there, benign. But the malignant one here, the idea behind it is why they are fixed to surroundings, that they like to invade, okay, and they like to infiltrate, okay, and they like to invade and infiltrate the surrounding tissue. But here they just like to press upon the surrounding tissues, the benign ones. So that's the concept. Now they malignant one, they don't respect any boundaries. They just send out irregular projections across the whole neighboring tissue, right? They destroy the capsule, even if they have any sort of capsule, and they just stick to the surrounding tissue. They adhere, right? And during this whole process, they stimulate something which is called as desmoplasia. Now, what is this desmoplasia? Desmo means dense, and plasia means growth. So a dense fibrous stromal reaction, right? Desmoplasia. It's like fibrous tissue, fibrotic tissue response around the invading cancer, makes the mass hard and fixed, right? So this is the mass, and here will be fibrosis, a lot of fibrotic tissue here. So that is desmoplasia in simple words. So desmoplasia, process by which the tumor becomes firm because it is neoplastic, and it induces fibroblasts within the stroma to form abundant collagen. Okay, now these collagen, right? We need this collagen to stabilize this malignant tumor. That's the concept behind it. Now, carcinoma in situ. What is carcinoma in situ? AKA high grade dysplasia. What will happen is that when dysplastic cells or dysplastic changes involve entire thickness of the epithelium, the whole epithelium, earliest form of epithelial malignancy. That's carcinoma in situ. So what I'm trying to understand and explain is that carcinoma in situ. What is carcinoma? Carcinoma is just cancer of gland. Okay, carcinoma. Okay, it's a malignant tumor, right? And it comes from three main tissues: ectoderm, endoderm, and mesoderm. Right? Ecto is like the outside the skin. It makes skin and gland. Endo means the inside, means like gut, lining gut, respiration, and mesoderm is the inside, the muscle one. Okay, so carcinoma, it from the epithelial tissue, right? And if the tumor comes from mesenchyma, we call it sarcoma. Okay, so the C in the carcinoma is the cancer of coverings, right? Think about like this: whenever your body is covered or lined, there is carcinoma, right? The carcinoma can arise. For example, skin covers us, right? Glandular organs like breast, right, prostate, they can give rise to carcinoma, right? So this is carcinoma. Okay, so cool shit. What other things? Cool things. Difference between malignant and benign. We understood it. We understand this carcinoma in situ, right? Well, this in situ, it really means that it's in its original place. That's it. In situ, in situ, in whatever you want to call it. It's like this pre-invasive malignant lesion, right? That means that the cell has become cancerous in its behavior and the DNA has really changed, right? But they have not yet invaded the basement membrane of this whole thing, right? So this is what I'm trying to say. Okay. Okay, so name the lineage of the following benign and malignant tumor. So these are the lineage I told you: epithelium, mesenchymal, lymphocyte, melanocytes, and adenoma and papilloma. Cool. So we have 1, 2, 3, 4, 5, 6, 7. So 7 lineages, right? So not 7, 6 lineages. First is epithelium. So epithelium lineage, right? Now here, every tumor has two parts, okay? Adenosarcoma, for example, adenocarcinoma. So this has one thing, for example, cell origin, right, and the nature of the cancer. Okay, that's benign or malignant. So adeno means gland, and oma means benign. Okay, adenoma, it's a benign cancer of gland. Okay, so if you know what tissue that tumor came from, whether it's benign or malignant, you can predict its name. That's what I'm trying to understand right there. So benign versus malignant naming system is done. It's like, for example, tissue of origin, right? Let's see. These are tissue of origin, right? So in mesenchyme, right, we have connective tissue, muscle, and bone, right? And these give rise to benign tumor name as lipoma, fibroma, right? Chondroma, osteoma, angioma, right? But in malignant type of mesenchyme origin, right? Mesenchyme origin in malignancy gives name liposarcoma, right? Angiosarcoma, chondrosarcoma, osteosarcoma, right? Now we have sarcoma almost in the end, right? In all. Melanocytes give us nevus, that's called mole. These are benign cancers. Now they are not dangerous, but we don't have something called a melanosarcoma. We have melanoma. Okay, that's really malignant in nature. Now, why we have oma in benign, right? It's a good question. It's like oma simply means tumor in Greek. So early pathologists name these name these lumps oma basically. So that's why we have these melanoma, osteoma, chondroma, angioma, except some of the exceptions, like malignant exceptions, which are melanoma, lymphoma, semioma. These are malignant. Now let's try to understand one by one. So adenoma means gland, right? Adenoma means gland, and oma means just benign tumor. So benign epithelial tumor that forms gland. Okay, thyroid adenoma is one of the example. Pituitary adenoma is one of the example. Adenoma can produce hormones functionally, and they can grow silently. Okay, and that's the concept for adenoma. Papilloma. Okay, papilloma is like papils means fingers, right, and oma means growth. So finger-like projection, growth. So benign epithelial tumor forming papillary finger-like projection, right. Examples can be skin cancer, right? Squamous papilloma, or maybe they can be a bladder cancer, for example, urothelial papilloma. Just think about it. It's like a growing out situation right here, like that. Okay. So this is papilloma of the skin or of a bladder. Lipoma, well, lipoma is fat, so benign tumor of adipose tissue. Again, angioma means heart blood vessels, so benign tumor of the blood vessels, angioma. Again, we can give another examples for this. Lymphoangioma, lymphatic vessels cancer, right? Chondroma, we know chondroblast, they exist, right? And chondro means cartilage and oma means cancer, so benign tumor of cartilage. It happens in small bones of hands, right? Osteoma, again, bones. Osteo means bone. Benign tumor of bone happens in skull or maybe in bony masses. Lymphomas. Now, why lymphomas, right here, has no benign form? Now it's a cool observation. I saw it. This is because the lymphoma tissue, like lymphoid tissue, is made up of free moving immune cells, right? We don't have like a one lymphoma tissue, right? We have free moving immune cells like B cells, T cells, natural killer cells, right? They're part of lymphoma system. They're part of the lymphoid tissue system. Now, they don't form any normal static mass, right? They keep moving, they keep circulate. So if they start proliferating abnormally, what will happen that we will have systemic malignant process across the whole body, and they will spread everywhere. So they will spread via lymph or blood, right? Thus, benign lymphoma doesn't exist because there is no benign discrete oma. There is no discrete malignant. There is no discrete formation of mass in this lymphoid tissue system. So these lymphoma are reactive lymphoid hyperplasias, right? And they are just cancers, right? So that was lymphoma. Okay, why epithelial tumors? They divide into adenoma and papilloma, right? Why they even do it? Because epithelium can organize into different architectures, right? Different type. Either epithelium, right? Epithelium can make glands, right? Or it can grow like a finger. That is papilloma. So they can make gland or papilloma, right? So cool shit. Why melanoma? Why the tumor of melanoma is not melanosarcoma? Why it's melanoma? Why melanocyte carcinogenic cancer gives rise to melanoma, not melanosarcoma? Why is it happening? Well, the thing here that these melanocytes, right? These are like neural crest cells, right? Neural crest derived cell, right? And they are not epithelium cells. They are not mesenchymal as well. Okay, they are like in the between. Okay, so they don't really fit into this classification of benign and malignant, right? They don't fit into this classification. I mean, yeah, I mean the benign part of melanocyte cancer is called mole, right? Not like oma, but it doesn't fit into this rule of carcinoma versus sarcoma rule, right? So what happened was that they named it melanoma, although it's malignant, but oma, this part stuck like for like some historical reasons, right? Because they just named it. I don't know why they named this melanoma. It sounds like it's like a misnomer, I think, right? So that's the whole thing right there. What is the leading cause of death in children, right? What is the leading cause of death in adults and in children? Okay, so in adults, number one is cardiovascular diseases. In children, is number one is accidents. So cool thing. Second is cancer. So both children and adults, they get cancer. And third is in adults, so cerebral vascular diseases, the brain diseases, and congenital defects. So as we can see, cancer is like persistent in both of the cases, right? What are the leading cancers by incidence and death toll in male and females? So cancer by incidence mean how much cancer is happening, right? So cancer by incidence in male is number one is prostate, right? But it doesn't kill you, right? The number one cancer that kills male is lungs. Lung cancer kills maximum people. And second is lung, right, in male by incidence. Third is colorectal. By death toll, the number one is lung, second is prostate, and third is colorectal. In female, the number one incident that happens, the number one type of cancer that happens is breast, second is lung, third is colorectal. In female, the number one killing cancer is lung again, second is breast, and third is colorectal. So colorectal is like one of the least, by incident. So here, cool thing is that lung has been number one killer of male and female from 20 years. And I read that, and I even heard that by the writer of Pathoma. He also told that. So that's cool point right there. Okay, lung cancer doesn't have an easy screening test like others. It's usually detected in the later stages. That's why it kills the maximum people. What are some properties in neoplasia? Okay, approximately after 30 division, we have neoplasia. Okay, so approximately 30 divisions before earliest symptom rises, right? So that's one property, cool thing. Subsequent divisions result in increased mutation, cool. Cancers don't produce symptoms till late. Example, ovarian cancer, pancreatic cancer, lung cancer, so important because lot of the space to expand, right? Lot of cavity to cover. So they will have accumulated tons of mutation, hence poor prognosis of these lung or these pancreas, right? What are the what what are what area goals of cancer screening? Why should we do it? Well, we must catch dysplasia, right, before it becomes carcinoma, because we know dysplasia is irreversible. I think I just told you what was dysplasia. It's like cells getting displaced, right? So cells getting displaced means, it's like dysplasia means that cells look really weird. They look really abnormal, and they grow in a very messy and disorganized way, right? But they're still in place, like they're getting displaced by some other kind of cells, right? So it's like disordered growth or abnormal development of the cells within that portion. Okay, so that is dysplasia. So we must catch that dysplasia before it's too late, because dysplasia is irreversible. Okay, think about it. Think about dysplasia as like a classroom setting. In normal tissue, students, right, the cells, they sit in good rows, neat rows, from small to tall. Everyone is behaving nicely. But in dysplasia, students are scattered everywhere. Some of them are very huge, some of them are very tiny, some of them facing the wall, some total disorder, total chaos. And they're still inside the classroom, still inside the basement membrane. Once they break the basement membrane, once they break the wall, the carcinoma, the true cancer, arises. So that's the concept of dysplasia. I hope I'm able to explain it to you very well. So catch the dysplasia before it's become carcinoma, because we don't want carcinoma. Detect the carcinoma before clinical symptom, because if the clinical symptoms are coming, it means we are too late. So these are like good screening test: pap smear, mammography, prostate specific antigen and DRE rectal exam, hemocult test, and colonoscopy. So pap smear, good for cervix, right? Cervical intraepithelial neoplasia. Mammography, great for breast cancers, ductal carcinoma in situ. These One of the problems with mammography is that they can actually get confused between these Ca deposit, calcium deposits in breast, so this can happen. Prostate specific antigen, prostate cancer usually grows on rectal side, right? Now rectal side, and does not produce any urinary syndrome until it's late, because it grows backwardly, like grows on the rectal side. Okay, it doesn't grow anteriorly, so the growth is posterior, right? And this BPH grows certainly and produce urinary syndrome, right? So BPH is another syndrome, and PSA is another thing, right? DRE is digital rectal exam. We need it, right? Hemocult test and colonoscopy. Hemocult test, colorectal cancer, detects blood in the stool. Hemocult test, right? Okay, so that was really, really like introductory video for neoplasia, and I hope we are able to understand this neoplasia very well and this naming scheme behind it.